Welcome to this lecture on the properties of alpha-amino acids. If you have ever wondered how the human body constructs complex structures like muscles, enzymes, and tissues, the answer begins right here. Today, we are going to explore these tiny molecular building blocks, understand their unique chemical behaviors, and see exactly why they are so essential to life itself. Let us dive in. To start our journey, imagine you are building a massive, complex castle out of Lego bricks. In the human body, proteins are these magnificent castles, and alpha-amino acids are the individual Lego bricks used to build them. However, these are not just ordinary plastic blocks. They are more like smart magnetic blocks that can actually change their electrical charge and physical shape depending on the environment they are placed in. Understanding how these tiny molecular bricks behave in water and react to acids and bases is the absolute first step to understanding how life is constructed at a molecular level. Now that we understand their role as building blocks, let us look at their actual structure. Formally, an alpha-amino acid is an organic molecule that contains both a basic amino group and an acidic carboxyl group. The key feature is that both of these groups are attached to the exact same central carbon atom, which we call the alpha carbon. Attached to this alpha carbon are four distinct components: the amino group, the carboxyl group, a simple hydrogen atom, and a unique side chain known as the R group. It is this specific arrangement that defines the molecule. Building on that structure, let us focus on the R group and the alpha carbon. It is this R group that makes each of the 20 standard amino acids completely different from one another, giving them unique sizes, shapes, and chemical behaviors. Because the central alpha carbon is bonded to four completely different groups in almost all amino acids, it acts as a chiral center. This means these molecules have a specific three-dimensional handedness, much like your left and right hands. This optical property is crucial for how they interact with other molecules in the biological world. Understanding their structure naturally leads us to ask why these specific properties matter so much in real life. First and foremost, their ability to act as both acids and bases allows them to function as vital buffers in our bloodstream. They prevent dangerous spikes or drops in our blood pH. If they did not have this dual nature, the slightest change in acidity from eating or exercising could be fatal. Furthermore, the unique properties of their R groups determine exactly how a long chain of amino acids will fold up into a functional three-dimensional protein, turning floppy strings into precise molecular machines. This buffering ability brings us to one of the most important properties of an alpha-amino acid, its acid-base behavior. Specifically, we need to talk about its ability to exist as a zwitterion. A zwitterion is a unique molecular state where the molecule has both a positive charge and a negative charge at the exact same time. Because these charges are equal and opposite, they perfectly cancel each other out, resulting in a net neutral charge for the entire molecule. This delicate balance is highly dependent on the acidity of the surrounding environment. To find exactly when this zwitterion state occurs, we use a specific mathematical formula. Conceptually, this formula finds the exact pH level, called the isoelectric point, where the amino acid reaches a perfectly balanced neutral state. Let us look at the symbols. The term Pi represents the isoelectric point itself. The term pKa1 represents the acid dissociation constant for the carboxyl group, which tells us how easily it gives up a proton. The term pKa2 represents the dissociation constant for the amino group, indicating how easily it accepts or releases a proton. By adding these two values and dividing by 2, we are simply calculating the mathematical midpoint. If the environment's pH drops below the pi, the molecule becomes positively charged, and if it rises above, it becomes negatively charged. Before we move on, let us see this formula in action with a concrete example. We are asked to calculate the isoelectric point for alanine, a simple neutral alpha-amino acid. We are given that the carboxyl group has a dissociation constant of 2.34, and the amino group has a constant of 9.69. Because alanine's R group is just a simple methyl group, it does not have its own charge, so we only need to average the main carboxyl and amino groups. We substitute our given values into the numerator and add them together, giving us 12.03. Finally, we divide by 2 to find the exact arithmetic mean. Our final answer is an isoelectric point of 6.015. This physically means that at a pH of exactly 6.015, alanine exists almost entirely as a neutral Zwitter ion. Now that we have calculated the isoelectric point for alanine, let us understand what this number actually means in a laboratory setting. As we found, at a pH of exactly 6.015, alanine exists as a neutral Zwitter ion with no net electrical charge. Because it is completely neutral, if you place it in an electrical field, it will not move towards either the positive or the negative electrode. It stays perfectly still. This specific physical property is heavily utilized by scientists to separate different amino acids from a mixture using a powerful laboratory technique called electrophoresis. This straightforward calculation works perfectly for neutral amino acids. But this brings us to a slightly more complex scenario. What happens when an amino acid has an extra acidic group in its side chain? Glutamic acid is a perfect example of this, as it possesses a third ionizable group. When an amino acid has an ionizable R group, we cannot just average the highest and lowest values like we did before. Instead, we must carefully select and average only the 2 specific dissociation constants that bracket the neutral Zwitter ion state. For an acidic amino acid, this neutral state occurs between the loss of the first and second acidic protons. To handle this third ionizable group, we use a modified version of our formula. Conceptually, because the neutral charge state for an acidic amino acid is bracketed by the 2 acidic groups, we completely ignore the high value of the basic amino group. Let us look at the updated symbols. The term Pi is still our isoelectric point. The term pKa1 remains the dissociation constant for the main carboxyl group. However, the second term is now pKar, which represents the dissociation constant for the side chain carboxyl group. By averaging just these 2 acidic constants, we ensure we are looking at the correct chemical transition window, where the net charge shifts from positive to neutral to negative. Now that we have our modified formula, let us apply it to glutamic acid. We are given 3 dissociation constants, 2.19 for the main carboxyl group, 4.25 for the side chain, and 9.67 for the main amino group. Because this is an acidic amino acid, our first crucial step is to completely ignore the high basic constant. We set up our formula using only the 2 acidic constants, substituting 2.19 and 4.25. Adding these together gives us a sum of 6.44 in the numerator. Dividing this sum by 2 reveals our midpoint. Our final answer is an isoelectric point of 3.22. This tells us exactly where glutamic acid becomes perfectly neutral. So we have successfully calculated the isoelectric point. But what does a value of 3.22 physically mean for glutamic acid? It means that this molecule is overall neutral only in a highly acidic environment, which makes perfect sense given its acidic side chain. But think about the human body. If this amino acid is placed in a neutral human bloodstream at a pH of 7.4, the environment is much more basic than its isoelectric point. Consequently, glutamic acid will carry a net negative charge in our blood. This permanent negative charge heavily influences how the resulting protein interacts with water and other molecules in the body. To visualize everything we have just learned, let us look at this diagram showing the zwitteron states at different pH levels. On the far left, in a low pH or highly acidic environment, the amino acid has accepted extra protons, giving it a net positive charge of +1. As we move to the center, we reach the isoelectric point. Here, the molecule has lost one proton, creating a state with both a positive and a negative charge that perfectly cancel out, leaving a net charge of 0. Finally, on the far right, in a high pH or basic environment, the molecule loses another proton, resulting in a net negative charge of -1. The arrows clearly show how adding or removing hydrogen ions drives these crucial transitions. We have covered a lot of ground today, so let us summarize the key takeaways. First, the properties of alpha-amino acids are fundamentally driven by their unique structure, featuring both acidic and basic groups on a single central carbon. Second, this dual nature allows them to act as zwitterions, giving them distinct isoelectric points that dictate their electrical charge in different environments. Finally, by mastering how to calculate and interpret these properties, you now understand the chemical foundation that dictates how proteins fold and function. You have taken a massive step in understanding the bedrock of biochemistry. Great job today.